me why really this bacterial system and how can we really extrapolate this example into the plant system, because these are completely two different systems, I can understand. But it is possible. It is possible, and Mittler in that preview paper, he actually proposed a model, I would say, that in that model, he actually showed plant can also overcome this situation or overcome this toxic environment by activating some functions or adapting some functions. For example, here you can see this, the ROS can be produced by signaling path from the signaling pathways and also metabolically or externally. But this ROS can be controlled or maintained by scavenging with different antioxidant mechanism. For example, superoxide dismutase, catalases, peroxidases, glutathione peroxidases, etc. And also, as I mentioned, in the bacterial system as well, the sequestration is very popular or very common phenomena, I would say. And so these are harmful ions and can be sequestered into the vacuoles, so that it will not really harm or it will not really initiate any production of ROS. And also, if some damage can happen or damage can already occur into the system, that could be repaired by the DNA and protein repair system, and also protein and organelle removal by autophagy, as I can say as an example, and also new biosynthesis. But there will be always some damage that cannot really, in the high toxic condition, you cannot really control all the cells to be damaged, but you can minimize the situation very well. So, but in most of the cases, you can see ROS leads to the signaling. So when it leads to the signaling, basically it initiates the downstream gene cascades to fight against the ROS. So which is very essential for the plant. So I would say, or it is better to say that because of the plant and animal cells have high ROS scavenging capacity, and the example that I presented here provide an evidence that reactive oxygen species, even at very high levels, cannot really kill cells. It is becoming more and more plausible that ROS-induced cell death is mediated through a programmed genetic pathway. So it can initiate it, but it is the programmed genetic pathway that leads to the cell death. So, and also it is not a result of the cell simply succumbing to an overwhelming ROS injury. Perhaps I would say, like, perhaps the emerging concept of redox biology that I will discuss in coming slides can provide more clarification to this question. So redox biology, what is redox biology and what can actually happen into this total system will answer our questions that we raised in this slide. So redox biology, if you ask me, or if I can try to actually define it, so it's a broad term used to describe the regulation of gene expression, metabolic pathways, and signal transduction mechanism by redox reactions mediated through the oxidation and reduction of cysteine residues of proteins that alter their structure and function. So here you can see that the paroxysm, mitochondria, chlorophyll, and cell-bound peroxidases result in the accumulation of H2O2, that means hydrogen peroxide or superoxide anion, into the cell, and that mediates the oxidation of cysteine residues into the protein, affecting their structure and function, and that triggers the cellular signaling pathways, here the signaling pathways. However, the presence of this labile iron, as I mentioned before, that it could be very detrimental or initiates a ROS, can change the situation in a way so that it leads to the production of hydroxyl radical. That will ultimately leads to the DNA damage, protein oxidation, lipid peroxidation, chemical cross-linking, etc. Ultimately produce the oxidative damage to the cell. So maintaining the cellular pool of labile iron, I would say here, as low as possible, therefore is very crucial for redox biology. And for the regulation of metabolism and other cellular function of ROS, this balance or this regulation is very, very important. So we will discuss this about in our coming slides also, but now I will give some examples as we discussing about the positive side of the reactive oxygen species. So I will give some examples where ROS plays a very specific roles in a positive way in plant growth and development. So there are emerging evidence on that ROS homeostasis control the plant vegetative apex form development. In this example is in Arabidopsis thaliana, where superoxide anion are basically accumulate into the meristematic region of the root tip and is required for cell division, whereas this hydrogen peroxide are basically important for the elongation zone and which confers the cell differentiation. These two different ROS microenvironments coincide with the meristematic and elongation zone, and their distribution are very important for the localization of the transition zone. Because of the ROS species gradient, cells entering to the transition zone can still proliferate. So once the ratio of superoxide anion to hydrogen peroxide reaches a certain level, cells stop dividing and begin to elongate. So therefore, the balance between this hydrogen peroxide to superoxide anion is very, very crucial, and it's important for the transition zone. So, so far we can say that it's really important for the ROS into the growth and development. It has also can trigger the plant organ morphogenesis. So the first example that I will talk about here regarding the seed germination. So in the dry condition and dormant seeds, plant embryos and the surrounding endosperm display very limited metabolic activities. So they are dry, so the metabolic activity are really, really low on that case. So ROS production, thus in the speculated there into the dry samples are very, very also very low. It is clearly, we can say that it's very low ROS production over there. But after the seed imbibition and during germination, metabolism rapidly resumes. So, and also the ROS production increased via various pathways. The spatiotemporal correlation of increased ROS production and accumulation during the onset of germination can positively influence the release of dormancy and also the germination. So ROS level increase after the seed imbibition. Yes, because it started, all the metabolic pathways has been started there, and also it acts as a positive signal for germination. However, I should mention here that above after certain limits, ROS are either too low to allow the germination or could be too high and affect the embryo viability and therefore prevent the delay or maybe sometimes delay the germination as well. So I would say the ROS homeostasis here during germination needs to be highly controlled or very tightly controlled. So here you can see there are some genes which are really involved into the pathway, and basically ABA and gibberellic acid pathway, they are responsible for the seed germination, and here ROS plays a very important role. On the other hand, I will talk about the floral morphology as well, because there are some reports where they have presented there that ROS has really important roles in case of the male gametophyte development, also female gametophyte development, sepal and petal formation, and also about the fertility. So here are the genes, those are responsible for male gametophyte development. Here are the genes for female gametophyte development and also petal and sepal development. So there are some genetic analysis, and it was observed that loss of function of any of these gene or some of this gene could be really detrimental and can also resulted into the abnormalities during the flower formation. So maybe there are problems with the male gametophyte development, also the female gametophyte development. Also there are loss of fertility that happen. Also pollen tube formation was being challenged because of the loss of these genes. So with this we can say that the plant organ morphogenesis is also somewhat dependent on the ROS trigger. The interplay between ROS and epigenetic modification landscape contributing to the stress. It is a very new topic to discuss, and also very few experiments and few papers are coming up regarding that. But this is very interesting. This has been very interesting in a way because the epigenetics are the new science now. So it's very important to know about it. So stress-inducing factors can modulate the level of ROS, which affect the epigenetic landscape by modulating DNA modification, histone modification, non-coding RNA, expression and also ATP-dependent chromatin remodeling. So this will subsequently affect the gene expression or gene expression patterns in the nucleus and mitochondria, which can contribute to the stress response. Although the underlying mechanism and consequences of reactive oxygen species in this ATP genetic modification are still not completely resolved, and there are much more studies needed to really comment on that. But still, it's a very interesting topic. In case of plant immunity and stomatal closure, there are some reports about the ROS production, ROS reactive on species. They can be really play some role there. So first example I would discuss about into a plant pathogen interaction between the fungal cell and into the plant cell. So Wang et al. Actually showed a very nice model of inter-kingdom ROS signaling, where in the interaction between a fungal pathogen and their host plant cell. Plant cells generate ROS in the plasma membrane and from several intracellular organelles upon pathogen recognition. So after it is attacked by the pathogen, they activate their this signaling pathways, and also it generates ROS into the system. In the meantime, in the fungal hyphae, produce ROS by then this NOx complex, mainly localized into the plasma membrane or endoplasmic reticulum, which stimulate an oxidative burst response within the pathogen. Scavenging system composed of enzymatic and non-enzymatic system synergistically function to maintain the intra and extracellular redox homeostasis in both plants and pathogen. On the other hand, if I talk about the stomatal movement, I'm not really happy about the picture, but you can see only a few of the genes which are responsible for the stomatal closure, but there are involvement of ROS here. So still I will try to explain it. Stomatal movements are basically controlled by complex signaling networks that respond to environmental and endogenous signals. So regulation of stomatal aperture requires coordinated activity between reactive oxygen species generating enzymes, signaling proteins, downstream executors such as ion pump, then transporters maybe, and also plasma membrane channels that control the gut cell target pressure. So accumulation of ROS in the apoplast and chloroplast is among the earliest hallmark of the stomatal closure. So when plant really sense and the ROS and it triggers the signal, so plants that basically close the stomata. So that is also a first signal of the stress that perceived by the plants. So, but even I must say that full understanding of the mechanism underlying between the stomatal closure and stomatal responses and plant pathogen interaction will be required because this is very, very early to really comment on that. But there are significant results or indication that reactive oxygen species play a significant role in plant immunity and stomatal closure. So far, I discussed about the positive sides of the ROS, that is when it really works as a signaling molecule, so it really contributes for them in a positive way. And that time, the accumulation of ROS is not that much high, so it works as a signaling molecule. But I should mention here that the scenario can be completely opposite in the situation where plants perceive or undergoes different abiotic and biotic stresses. So probably, and I hope that we all know what is abiotic and biotic stresses are, but in general, heat, light, drought, any kind of physical stress, I would say, those are related to the abiotic stress. On the other hand, the biotic stress really need an organism and any pathogen or something that can really harm the plant physically, and that is related to the biotic stress. So what happened into the abiotic and biotic stresses? So mostly, as I mentioned, the photosynthetic machinery and accumulation of the ROS into the photosynthetic machinery harm in case of the abiotic stress. Also elevated transpiration and decrease of carbon dioxide fixation, cell membrane integrity, lip senescence, increased metal concentration, hormonal imbalance, then reduced nodulation and nitrogen fixation, and some other options are also there. So in case of biotic stress, the symptoms or maybe the effects are a bit different, but ultimately they will suffer the same kind of effect. So Reduce shoot development, you will see, then mostly susceptibility to the disease, damage photosystem and reduce photosynthesis, then also inhibit the root growth and impaired cell division, then reduce plant biomass, hormonal imbalance, etc. So there is this cartoon or this figure, I would say, is very famous into the stress biology because it is very simple, but yet very descriptive to show the actual scenario into the plant system. So here you can see in the normal condition, the antioxidant and ROS, they are in a equal control. So there is a equilibrium presence between the antioxidant and oxidants into the plant cell. So it is an redox equilibrium is present. But in the oxidative stress condition, or the problems occur when the concentration or the amount of ROS accumulation increase into the plant cell. So this equilibrium that breaks and it causes the oxidative stress. This can happen also in the case of depletion of antioxidants. So it will harm in the same way. That means the equilibrium can be imbalanced by either excess of ROS generation and also depletion of the antioxidants into the system. So it is really, really important to get the balance right. So the equilibrium to the plants should be very balanced so that plants really can see reactive oxygen reactive oxygen species will really generate into the plant system very naturally. So it will generate the way, but the antioxidant system should be very effective over there so that it can really scavenge it. But under the stress condition, it is not the normal scenario. So plants cannot really scavenge always. So then the equilibrium or the balance is not in the right condition. So it deteriorates. Later, I will really describe about the direct and indirect ROS effects. So mostly during the abiotic and biotic stresses, sensors, plants or cells basically undergoes severe effects on lipid peroxidation and DNA damages and others. So here you can see it can also change the epigenetics. In case of the DNA, so many things can affect. For example, deoxyribose oxidation, strand breakage, indels can happen. Then depurination or depyrimidination can happen. Protein DNA crosslinks can happen, etc. These all the phenomena will lead to the chromosomal aberration and also mutations. Ploidy level can also be changed. And also in the protein level, you can see the possible loss or gain of protein function can happen. So it will disturb in the cell cytoskeleton and mutic and mitotic disorders. And also it can modify the enzymatic activity, alter the membrane selectivity, and also change sensing and also the signaling system as well. So although at a first glance, you will see that plants only harm the lipid peroxidation and also the protein oxidation, or maybe at the DNA level, but the direct and indirect cross-induced effects are so many and so huge. So I would say, like in every steps of the plant cell, they can really sense the level of this effect. So there are some defense machinery itself into the plant cell because plant has an inbuilt antioxidant network into the system itself to scavenge this situation. So this antioxidant network is very important and mostly divided into two types. Those are enzymatic components and non-enzymatic components. So enzymatic components are basically superoxide dismutase catalysis, APX, that is ascorbate glutathione ascorbate peroxidase, glutathione reductase, then GSTs, then glutathione peroxidase, etc. On the other hand, non-enzymatic components are ascorbate, glutathione, then tocopherols, carotenoids, and cysteine, sugars, prolines, etc. So, but in today's webinar, I will basically talk about some of the enzymatic components, those showed already into the plants or into the crop system, that they can really affect or they can defend plants in case of reactive oxygen species environment. So if we see the distribution of the main oxidant resources into the plant cell, they are mostly into the main organelles, chloroplast, cytosol, vacuole, the mitochondrion, and peroxisome nucleus. It is very noteworthy to mention here that you can see that as we discussed when the source of the ROS, we discussed about chloroplast, then mitochondrion, the sparksism, and even at the cytoplasm. So where are, which are the source of reactive oxygen species is also have the defense mechanism inside to minimize that. So plant itself have the machinery to overcome the situation. Just it needed the right balance over there. So if I discuss about the ROS scavenging antioxidant defense mechanism, there are 4 different types of cycles. So water cycle, where actually sodium dismutase is the main component. Then it has the GPX cycle, where glutathione peroxidase and glutathione reductase works as the major, play the key role over there. Then ascorbic glutathione pathway is very famous for as defending the plant system from the ROS, where glutathione peroxidase, ascorbic peroxidase plays the important role, and also the catalysis. It has very important role there. So what really happened when plant undergoes the stress? So first they perceive these signals, or this sense the signal there are some stress and the unfavorable condition. So the osmotic stress or oxidative stress really happen, and the signal sensing, perception, and translation situation occur. So it activates the downstream transcription factors, and the transcriptional control activates the gene activation downstreams. And ultimately, all the gene cascades, those are working to, in the defense mechanism, they are activated in the sense, and at the end, they all together produce or make a mechanism so that the plant can tolerate the stress or give some resistance against the stress. So there are so many papers, and it is the transgenic approach to overexpress some genes that can really give some resistance in case of the stress conditions are available here. So there are so many papers. Maybe this slide is not very clear to understand, but here you can see so many genes, for example, SODase, APXS, catalysis, then also DHR, glutathione reductase, GPXS. These are overexpressed in different plant systems to minimize the ROS condition. So I will discuss about some examples. So for example, in 2016, Fenatol showed some data regarding the SOD into the tomato, and from the expression profile, you can see here that the superoxide dismutase are really expressed highly into the different plant tissues. And also under the stress condition, in the salt and drought conditions, they are highly expressive in some cases. And from the genome-wide in silico analysis, there are basically 3 types of SODs. That means superoxide dismutase, these are iron type, then manganese type, and copper zinc SODs present in the plant system. This gene, that means in the they are also overexpressed in rice, and they showed better performance, I would say, in the, if you see here, the germination rate and also the fresh weights, then MGA content, and also the plant height, everything. That means all the parameters that are required for to really analyze whether the plants are performing better or not. They showed better performance in case of in comparison to the wild type. So transgenic lines are really good in overcoming situation, and also in case of not only this salinity stress or drought stress, in cadmium stress also, you can see the transgenic lines mostly perform very better when the superoxide